Yes, good afternoon, everyone. We will discuss a few things today that are important for your assignment as well. So, I want to remind you all that I don't want you to miss the classes, especially because everything that is discussed here would always be reflecting on how well you are going to do the assignment. Okay, so a majority of the things are related to assignment. We are working our way through completion to the completion of the course. So in today's lecture, we will discuss a few things about PCR, not whole lot of details, but I think if you already know PCR, this would be fundamental, but we will discuss a few things. So first thing is that the DNA polymerases you have in eukaryotes, you may have in prokaryotes, E. coli ones. Majority of the ones that we employ in as an application, as a tool in recombinant DNA technology or genetic engineering is based on prokaryotes, bacterial DNA pol. And different organisms have different polymerases isolated from different bacteria have different properties, and we need to look into them a little bit carefully, and then we will probably go into little more details of it. There are several subunits. In some cases you may have only one subunit, in some cases you may have 7 or more subunits. Before I forget, I will write somewhere here. Translation DNA synthesis. Do you know about it? So, and I would also try to take a little bit philosophical in tone when I'm trying to describe this. There are 2 important properties of DNA polymerases. One thing which is essential for life. First one is that it has to polymerize, right? It has to polymerize, not just like that. It has to be dependent on a template. Because there is a DNA molecule, and we would want another DNA molecule to form with exact sequence. We do not want to have something different. If that was the case, then we wouldn't be existing as we are speaking now. In the sense, say for example, if this is the shape and it replicated and formed a different shape like this, then nothing of this would have been understood. Maybe I'll try to take up those things later. But what I mean to say is there should be stability in the way things are propagating. This is the time, for example, on a time scale. If you're trying to see now this shape is giving rise to different shapes, and like this. And what I will say, okay, like this, and a perfect this one. Well, at time point 0, this is the entity that was there. At time point 1, that has gone off. This is dead, and now you have a different one. And at entity 2, you have something else. There is no stability, and no entity can exist on a time scale if there is no fidelity. Fidelity, or in the sense to be able to replicate what is already existing. So there should be polymerization, and it should be template dependent. Only then we would have something like this. This will give rise to 2 new ones with the same things, almost same. There cannot be 2 identities exactly the same things. You will have different ones. Now at time point 2, you still have at time point 2, you still have the organisms like this. So I mean the entities are existing, right? So you need to replicate. You need polymerization. To replicate the same to have the same identity, you need to have template dependency, right? I hope you remember we spoke about a polymerase which is template independent. Can you recollect which one it is? It is terminal transferase, right? And terminal transferase is a DNA polymerase. It's a kind of DNA polymerase, but it is template independent. Whereas majority of the DNA polymerases that are involved in replication are all template dependent. No question about that. And so these two are important for polymerization and maintaining the identity of any of the DNA molecule as we are discussing. Then uh, um, then we have one more issue is that change is imminent, right? Have you seen Ratatouille? There is something a statement that like I think the father rat tells to the baby something like you can't change the nature. The rat replies, no, not that. It says change is change is nature. Okay, so there is going to be change, and this change will happen irrespective of however much we want to we take care of. There is only one difference. Do you want more change or less change? I want you to compare with these two scenarios. Okay, I understand we are going a little off the track in a philosophical way, but see if you can try to make sense of it, because the essence of life and also evolution, existence and evolution, is dependent on such kinds of thought processes. I would also encourage you to go to YouTube. There are several videos or playlists on philosophy. I think there should not be any scientist who is not a philosopher. You can have a philosopher who is not a scientist, that's all fine. But every scientist should be a philosopher. We should have some inclination to philosophy because it's about truth and we are trying to seek truth. Here I have situation A and here is situation B. Do you see any difference in the outcomes of it? In one case, because we are not following template dependency and change is very high, then we don't have, each of it is something else. It's not like the identity that it started out to be. Whereas if we can minimize the change, then the identity is maintained to to an extent, even after a time period. Okay, so you need polymerization and template dependency. What if there is also one more important thing that we need is the ability to repair. Okay, that means if a mistake has happened when we are trying to polymerize, say I'm trying to be here now. If I'm trying to polymerize and there is a mistake happened, and if I have the ability to repair this, then I will maintain the identity, right? So all of in the existence or evolution of living organisms, these things will become very important, where you need to be able to replicate. It should be template dependent so that the information is carried almost similar. And if there are changes, you should be able to repair. Those are the things that will make, and that's why DNA polymerases are at the center of all of this propagation. So we have three DNA polymerases, poly A, B, and C. I want you to notice that polymerization is always 5' to 3', and there you have two different exonuclease activities. One is 3' to 5' exonuclease activity. That is, if a DNA molecule is polymerizing here, it's adding new bases here, and say for example it has added a wrong base. Now the DNA polymerase is somewhere here, okay, somewhere here. It should track back and remove this A, the adenine that is here. And if it can do that, then it will be able to clean up that mess like that and then redo a proper base, unlike this. So it is moving, it was at 3, the exonuclease activity is 3' to 5'. So therefore this one, this activity 3' to 5' exonuclease activity is referred to as the proofreading exonuclease activity. There are also some other DNA polymerases such as DNA polymerase 1. It has 5' to 3' exonuclease activity. I think you may have studied. In 1 second. I'll try to. Okay, you may have studied in Nick translation something called as. You have a DNA molecule, and I think it's better to take something. Okay, let's do it this way. So you have 5', 3', 3, and 5. So if there is DNA polymerase 1, DNA pol 1 has a special feature. It is now polymerizing in this direction, as we are showing here. It is polymerizing, but here is a DNA molecule like this. And then what it can do is it can erase these parts slowly. I mean, it will chop off DNA, and then it will keep on synthesizing newer DNA like this. Especially when there are nicks, and if there are nicks, if there is a DNA molecule and exactly one, what is the difference between nick and a space? You can think of it like that. Is all the bases are present here and base paired. Only the phosphate backbone is broken. That's called a nick. Nick can be closed by or sealed by ligases. But if you have something like this, a DNA molecule with a space like that, DNA ligase cannot seal this gap because there are additional bases here. And even there is, in that case, one of the ways it can do is DNA polymerase is also involved in repair mechanisms. So what it can do is come here and start synthesizing DNA from here on like this. And because there is DNA here, it will try to, it can erase these parts, or I mean, it will chop off this DNA. And while it is chopping off, it will continuously synthesize the DNA molecule. That's called as nick translation. Okay. And at one point, it will become a place like this, and ligase will act upon it and seal the gap. So this polymerase is this exonuclease activity is DNA polymerase is chopping the DNA from here like this. So it is moving from 5' to 3' direction, and that's why you call it as 5' to 3' exonuclease activity. That is this one. There is one more thing we need to also discuss is polymerization rate. How many bases can it add? DNA polymerase is slow compared to something like polymerase Pol3. Pol3 is the one that is involved in replication. It has to work very fast. It adds up to 1,000 bases per second. That means, imagine in a second 1,000 bases are polymerized. That's so dramatic actually. And there is also, I hope you know what is processivity. That is number of nucleotides added before polymerase dissociates from the DNA. Say this is the DNA, and DNA polymerase is adding. And if there is a, if it falls off after adding, if this DNA polymerase falls off or dissociates from the primer junction template after adding 10 bases, whereas another DNA polymerase is there, it only dissociates after adding 100 bases, then the second one has higher processivity compared to that of the lower one. So here it means before polymerase dissociates, it adds between or up to 200 bases. whereas type 2 is 1500, and type 3 is, you can see, 5 lakh bases before it dissociates from the primer template. Uh, can you tell me why polymerase DNA pol 3 has higher processivity? Yes, if you remember, if this is the DNA polymerase is somewhere here, it is synthesizing, and there is some ring-like structure. What is that called as? Yes?
ORI. Beta clamp.
Yes, it's called as sliding clamp. That's right. Sliding clamp is the protein ring-like structure that binds around the DNA, and it also interacts with the DNA polymerase. So, kind of, DNA polymerase is held to the DNA being polymerized or replicated. Therefore, DNA polymerase 3 has very high processivity. Do you know what is the name of it in eukaryotes? Sliding clamp. Yes. Always try to remember, associate the term with the function. Proliferating cell nuclear antigen. The cell is proliferating. That means replication is happening almost continuously, and in that there is a nuclear protein that is usual. The sliding clamp of eukaryotes is referred to as pCNA. Okay. So I hope you understood the functions or structures of these functional aspects of DNA polymerases. And one thing I would want to discuss is how do you relate speed and fidelity? If I'm trying to do something very fast, very fast beyond my capacity, okay, if speed is high, then fidelity is going to go down. That means I'm prone to making more mistakes, right? If I'm slow, then fidelity is likely to be very high. What if there is a situation like I was supposed to describe about this one? What is translation DNA synthesis? That is the question I had. I'll try to discuss that now. Yes. So what if I have, there are some odd cases where speed is very low and fidelity is also very low. So I'll give you three situations. One is this, second is this, and third is this one. High speed, low fidelity, low speed, high fidelity, and low speed and low fidelity. Three situations I gave. What do you think? Which one is better, and what would be your choice in that? Yes?
High speed, low fidelity,
Why is that?
because it can correct it later even if there are mistakes. But speed is important, right?
Yeah, you tell your opinion. I'll agree or disagree. That's right. I agree with you. Why is second not okay? Or why is third not okay? So high speed, high fidelity, that's a very valid point, that there is a repair can be made. Okay. Why is speed even important? Right? It is like we do take approaches. I want to do it perfectly. That's why it is taking so long time. That's actually not a good evolutionary strategy. This is the running race or something, or deadline is this. You are doing it. You want to do utmost, you don't want to do any mistakes at all. So you are doing very slowly. By the time the race is over, race is not only in terms of length, it is also about, it is not only space, it's also about time. Okay. So there is only one way of not doing any mistakes at all. The mistakes would be to not to do anything at all. If we are going to perform if you're going to do something, there will be mistakes. It doesn't mean that one has to be reckless about it, but we need to ensure that we are doing it such a way that some errors are tolerated. If we want to be a perfectionist, then we are going to be very slow. And in a field of, say, for some example, for example, where there are 5 different bacteria, and one is saying, I will do it very slow and perfectly. By the time, everything else will multiply. All the resources that are there are no longer available, and this is going to die out. Okay? If you think in terms of evolutionary strategies, everything matters in terms of time, space, matter, and energy. Those are very expensive commodities, resources. One cannot look for ideal conditions, okay, ideal replication. I hope you understood what is the bacterial situation. That's how organisms do work. So here you have speed and repair. That's what normally happens. If you have low speed and high fidelity, it's very good to do, but it will fail. What about this one? No speed, no fidelity. That seems absolutely useless. But actually, there is some use to it in the in if you see in microbiology or molecular biology. If you read it, you might have found that. So what happens is, if there is a DNA and there are thymidine dimers. What are thymidine dimers, and when do they form? Thymidine dimers. Anybody? UV light. Okay, when due to UV radiation, thymidine dimers form. They form a cyclobutane ring. And assume that DNA polymerase with high speed and little lower fidelity, it still has fidelity. It is going to polymerize this. And these thymidines that are present in this location, they cannot make a base pairing. But all these are template dependent, right? The DNA polymerase 1, 2, 3, they're all template dependent. But then they don't know what to add here. In a way, I'm just trying to anthropomorphize, but just assume that they cannot add a base here, there, so they get stuck. At one point, because they are not able to add, they will fall off. And if the other one comes, like with low fidelity, low speed and high fidelity, it also has the same problem. It cannot add anything there because these thymidine rings do not base pair. Then comes this one. Very slow speed, low processivity, speed, as well as fidelity. It doesn't have fidelity. What it does is, it kinds of, there's a polymerase, I think DNA polymerase 4 and 5, they are involved in some kind of this kinds of activity. They'll come here and they will just add whatever is available. And because their processivity is also low, they will fall off. Now you have a 3' OH, and the DNA polymerase of this kind can come over and start replication, continue the replication. There is one more important thing that we need to learn from this is, or I learned from this. Do you see the need for this one? Do you see how something that is very low in processivity, speed, and fidelity is useful to the cell? If this replication did not happen, cell would have died. If this is called as translation DNA synthesis. Okay, this is the lesion, and you are synthesizing across the lesion, so you call it as translation DNA synthesis, and that is done by polymerases that do not have any fidelity at all. They just add, okay, whatever is here, they will just add something here and fall off. And the remaining work is done by the DNA polymerase 1, 3, and so on. So these are lifesavers in these kinds of situations. If you, but there is a problem, of course, that The added bases are all mutations, or if unrepaired, they would result in mutation. It is the strategy of life in biology is that it is better. I think I do ask in many of my classes is something is better than nothing, or nothing is better than nonsense. What do you think? What applies to you? So either of these strategies should be useful to us, right? Something is better than nothing, or nothing is better than nonsense. In biology, if you see, in all of biology that I know of, something is better than nothing. If it is nothing is synthesized, death. Something, even if it is wrong basis, is better because it gives hope for the cell so that it can later repair these things and it is still existing. Okay, so I think one should see the DNA polymerases, study them properly in terms of philosophy as well. It will be very helpful. You can type your questions. I will get back to this. Okay. So in PCR, all this what we are discussing is about polymerases using in PCR. There are several types, like um, Tech polymerase is the one. They are all thermostable. Why is that? Because we are going to use 4 different cycle of PCR. The cycle is initial denaturation, right? And initial denaturation, is around 95 degrees centigrade. And then you have the denaturation. That's about, it depends on the primer annealing temperature. Assume it is somewhere around this. And then we have extension. That's around 65 to 72 degrees centigrade. It depends again what polymerase we have used. Different polymerases have different optimal temperatures. And then we say go to step. Is that right? Denaturation? No. You didn't tell me. What is that? Annealing. Annealing is, annealing is 55 degrees centigrade. Then you have extension. That's around 65 to 72 degrees centigrade or so. And then we have final or go to step. We will repeat these cycles for 30 times or so. Then you have final extension, right? So we are using very high temperatures as these, which is not, if we used anything that of polymerase that of E. coli or so, they would have gotten denatured here. So we need to, that is why people have isolated different DNA polymerases from different thermophiles, because their enzymes are already naturally evolved or to withstand high temperatures. So they have taken DNA polymerases and used those things. Why I'm talking about this particular table is that you have different types of DNA polymerases. There is also PFX, but there are 2 enzymes I would want you to compare. One of it is PFU, and this is Taq polymerase. Taq polymerase has a mistake, has error rate, higher error rate, because it does not have. I would want you to watch this one, 3' to 5' exonuclease activity. That is, the proofreading exonuclease activity is not present. Okay, some others do not have 5' to 3' exonuclease activity, and some do not have also 3' to 5' exonuclease activity. Compare that with that of PFU. it has the 3' to 5' exonuclease activity. So those enzymes that have 3' to 5' exonuclease activity or proofreading activity are important or good for using in PCR. Okay. And you should also notice this is the extension rate. You can continue, assume this to be fidelity and this to be speed. There is no fidelity or exonuclease activity or proofreading activity. Its speed is somewhere around 75 nucleotides per second. And if you see that of where is it? PFU, usually it will be there, but it's usually less, something like this. It goes very slower. Okay. Um, in the sense, in the previous one, what we have discussed, speed and fidelity. Here, if you choose something that is very fast, very fast, it might make mistakes. So if you am cloning a gene and it has ATG and several codons and TGA, okay, and by mistake in the first round or so, it made a mistake here to make it as GTG or CTG, TGA, you will never get a protein upon expression. So one has to choose why you are doing the PCR. In some PCRs, you just do it for not non-cloning purposes. We will see in the several other discussions, we will see what are other non-cloning purposes we have in this one. For analysis purposes, we can do that. But if you want to clone, it is always better to use something of high fidelity. There are several others available. Okay. So Taq polymerase is not ideal. If there is nothing else, it's okay to use Taq polymerase. But otherwise, paying attention to proofreading capacity of the polymerase is important anytime we are thinking about cloning a gene. Okay, just keep that in mind. So PCR, I think majority of you have already studied. I will quickly go through this. In the first is you will have. So the first is this, that is you'll use the template. All the things that you have to add are template. Then we need to have primers. There will be two. You can most people call it as forward and reverse primers. Then you would also need DNTPs, and you will need buffer for which in which the DNA polymerase acts properly, and you will make it up with water. Okay, anything else is missing? Template, primers, DNTPs, buffer, and DNA polymerase. Sometimes we will need MgCl2, magnesium ions, for the functioning of DNA polymerase. Now, previously they used to add it separately. Now it is already, many cases, it's already incorporated within the buffer. So you don't need to add in additionally. So the cycle goes at 94 to 95 degrees centigrade, so that the template strands will separate, something like this. And then you will go to annealing temperature somewhere around 55 degrees centigrade. It depends on the melting temperature of the primers. Please find your own formula for finding the melting temperature of primers, because when you submit your assignment, I want you to, because you're going to design your primers, you will have to calculate the melting temperature and use it in your PCR protocol. Whenever you're going to mention that I'm going to go clone like this, then you are going to use PCR. If you're going to use PCR, I would want you to mention the specific details pertaining to your gene of interest or the gene in question for unique sequence that you got. And Then denaturation, this is annealing. And then we have another step is called as extension. That the temperature is dependent on the type of DNA polymerase that you're using. Somewhere different polymerases have different temperatures. It will depend something like this. I think, if I remember correctly, for the high fidelity PCR reactions where you're using a high fidelity PCR polymerase, you usually have higher temperature, but we can check that out. And then we are going to have several cycles, somewhere around 30 cycles, and keep on repeating, right? PCR, that way it is a simple technique. I'll post some animations if you haven't ever watched before, but I think majority of you have done this before. It's a quick review, and I'm trying to focus on the critical details. And now I'm going to, I think I have 20 minutes. I'm going to explain something about the assignment. I will send this document to you once I have finished all further more details. So you're going to clone in at least 5 different ways. I'm telling you now so that you will be able to contemplate or think about those things. How is it possible, and so on, right? You have several of them are directional cloning. Okay, I think this is the only one that is non-directional. I will decide later. So one is very simple, using restriction enzymes and ligases. Okay, a vector for each case will be described. Based on that, you will have to do. A vector might be given with one restriction site or multiple restriction sites, and then you will have to do the cloning using restriction enzymes, trying to find compatibility and other things. If there is no compatibility, then you may use linkers. In the second one, you have to do blunt end ligation, where that is where you're trying to use you're trying to polish either by extension using Kleinow fragment or using exonuclease. You're cleaving it and polishing the ends, and you're going to do blunt end ligation. Okay. Then there is one more. You have to do directional cloning using adapters. This is a trick. It is a question that we haven't discussed. We know about adapters and so on. I am asking you to see how you can add an adapter, for example, here BAMH1, and an adapter for EcoR1 on these sides. How can you do that? You can do however you will do it, but I want you to add adapters, one on EcoR1 and on the other side BAMH1. Why I'm asking this is I will tell you more details about what exactly. But if I took a DNA fragment, okay, if I took a DNA fragment like this, and I added, I want to add EcoR1 on one side. So I have taken the adapters and I added it. What will happen then? You shouldn't assume that it will just add on this side alone, and it will not add on this side. That's not the case. This is a very open-ended question. Different people have different strategies, and I am expecting that you will all try to come up with different solutions. There are multiple solutions to this. Say if I add like this, what will happen? And if this is the EcoR1 adapter, then you will end up adding on both sides EcoR1. I don't want that. At the end, the product I want is, on one side I want EcoR1, on the other side I want BAMH1. So the adapters are not absolute. At this point, I want you to think of how you can do those stuff. Okay? And in this case, in the first one, you might use linkers, you might use methylases. If nothing is possible, you might also, if nothing else is possible, you might also use homopolymer tailing. Okay. Yes, I explained this one. I explained this. So I'm giving you these insights so that you'll already have an idea about what you are supposed to do. Please think about these questions and try to imagine the answers. And once I will post it today or tomorrow, and once that is there, you will shall try to work it out. Then this is your self-study kind of thing. I will describe about topocloning only after I finish it. I was trying to add some resources. Maybe once you see the resources, you will get a better understanding. At the end, I want you to, as I was telling in the last class, describe and justify the best strategy. In the sense you are trying to evaluate which one is most feasible, which one is easiest, or which one is economical. In those ways, you should be able to explain. And once you do that, I think you'll have a better appreciation of all the things that you're learning from this course. Okay. Any questions? I would like to take questions now. Yes, please. We have 15 minutes. Hello? Any questions? Yes, please. If you don't have any questions, then I'll continue with another topic, a small topic. Let me check if I've finished everything. No, topoisomerase is after. I hope you can see this figure, right? I hope you remember this. I'll try to quickly go. I forgot to mention this one. During polymerization, this is the template, and this is the primer. Something like primer and template, you call that a primer template junction. So that is the substrate for DNA polymerase. And what else is substrate? You will need dNTPs. If you are talking about transcription, don't you should write NTPs, that is ribonucleotide triphosphates, nucleoside triphosphates. Here it is deoxynucleotide triphosphates. So be specific and clear about what you're writing and how you're writing it. And this is the in the incoming nucleotide is shown here, and that would have been the first one is alpha phosphate, this is beta, and this one is gamma. And the 3' OH of the primer makes a nucleophilic attack with the alpha phosphate and makes a phosphodiester bond there. And the beta and gamma phosphates are released out, which are in replication. Pyrophosphatase would have broken that, and it would have formed two inorganic phosphates. And now you already the incoming base has been added. That is the polymerization chemistry of polymerization. You will have more and more bases added as long as you have the template and the nucleotides to be added are around. Okay. Yes, I'm done with today's thing. I will next from tomorrow onwards we will discuss about vectors. Okay. So I would want you to ask me if you have any questions so that I'll try to answer. No questions?
No, sir.
I thought you're going to ask a question.
Sorry, sir.
Okay. I'll just check if I have missed anything.
Uh, should we submit by 10th of September, sir?
Yes, thank
Or
you.
is there any extension?
Thank you. Yeah. Uh, I want you to, of all these things, tell me which one is there anything that is not possible? You also can have TA cloning in this one. Okay. So here you have a lot of liberty about how you're trying to do. Please understand this. Once you try to go through this assignment, your ability to use these things in real life, I mean, when you are in the lab, for example, I'm trying to expand the way in which you can think, how many ways you can think. Okay, there is no one correct answer for for majority of the things. You have many others. If you try to know why all those are answers and if they are correct, then we have expanded our thought processes and we are actually improving our skills in thinking. It's all about the speed at which speed and spontaneity of thinking is what is required. For that, one has to practice diverse possibilities. One has to practice looking at diverse possibilities and picking out the best of the solutions. This assignment represents that. You have four or five different possibilities that you're looking at. You're working on your real life, a real problem that is sequence that is given to you, and you're also describing or justifying the best strategy, why it is the best one compared to 1, 2, 3, and 4 and 5, so on. And while you do that, you have a good amount of thinking for RDN, at least in that case. Okay. Any other questions, please? Oh, you didn't raise any question, right? Mm, anybody?